Good afternoon all. Am I audible? Rakesh sir, voice is clear? Hello? Today topic is photosynthesis. Today topic is photosynthesis. What is photosynthesis? Photo means light. Synthesis means synthesis food material. Light, presence of light, the plants can prepare their own food material. This process called as photosynthesis. In photosynthesis, the mainly depends carbon dioxide, atmospheric carbon dioxide, and water molecules in the presence of chlorophyll, sunlight, chlorophyll, and sunlight. This is light. As a result, formation of food material. Right? Right. Food material that is called C6H12O6 and release carbon dioxide. This whole process called as photosynthesis. The first photo light. So in photosynthesis, two stages are there. One is light reaction, another one is dark reaction. In light reaction, only assimilatory power form in the form of ATP, NADPH. The ATP, NADPH utilize carbon dioxide, atmospheric carbon dioxide, and water molecules. The combined, the form of energy, ATP energy and NADPH energy. This energy utilized for the carbon dioxide, water molecules combination. As a result, formation of carbohydrate. In this process, release carbohydrate. In this process, release carbon dioxide. Release into the atmosphere. So this is carried out by two stages. One is light reaction, another one is dark reaction. Right? Before that, we should know about the structure of chloroplast, because this entire process takes place inside the chloroplast. So we should know about the chloroplast, right? So chloroplast structure. Chloroplast mainly formed inside the leaf. These are developed. These are present inside the leaf. These are biconchave structure, double membrane, one and two membrane layer. So this is inner membrane, white color, intermembrane space between outer membrane. This is outer membrane, this membrane outer, and this is inner. Between the outer and inner membrane space, intermembrane space. This is intermembrane space. So this is the membrane layer. So membrane is double layer, right? Inside the chloroplast, stroma present. This empty place come together called as stroma. In stroma, the disc-like structures as a form of row form this group called as thylakoids. These are called thylakoids. This group of thylakoids called as granum. Group of thylakoids called as granum. The entire granum called as granum. So inside the chloroplast, some chloroplast enzymes are there, and chloroplast DNA also present, and ribosomes, RNA also present inside the chloroplast. So this is the clear view of chloroplast. And inside the thylakoid membrane, pigments also present. What are the pigments? Let's see some pigments here. The pigments are main pigments chlorophyll A and chlorophyll B. Apart from that, some auxiliary pigments also there. They are carotenoids such as keratin and xanthophylls. The phycobilins, phycoerythrin, phycoenzyme, they are present inside the algal membrane, not in higher plants. Right? In higher plants, mainly chlorophyll A and chlorophyll B. But in lower plants, such as algal member and bryophyta, apart from the chlorophyll A, actually this chlorophyll A is the universal pigment because it is present in any autotrophic organism. Right? So that's why it is considered as a universal pigment. Apart from the chlorophyll A, the extra chlorophylls also present such as chlorophyll B, C, and chlorophyll D and chlorophyll E also present in some algal member. Apart from that, bacteria chlorophyll A, bacteria chlorophyll B also present. So these are according to Arnon, total 9 different types of chlorophylls present. Among this, only the chlorophyll A and B, these both are very, very important. Right? And extra pigments called as auxiliary pigments. Actually, these auxiliary pigments not directly involved in photosynthesis. Here, the photosynthetically active pigments are chlorophyll A and B. But these auxiliary pigments assisting in formation of photosynthesis. These auxiliary pigments surrounds the chlorophyll A and B, and these auxiliary pigment first receive the light. And this light through the resonance process, the slowly gives to the reaction center chlorophyll A. Right? These carotenoids are keratin and xanthophyll. And the another pigment phycobilins, but which is present only in algal member, such as cyanophysin member and rhodophysin. These phycobilins are phycoerythrin and phycocyanin. Right? Let's see the chlorophyll structure. If we see the chlorophyll structure, in center magnesium atom present, and it's surrounded by 4 chiral rings. 1, 2, 3, and 4. This 4 chiral rings, and center, this is 1, this is 2, N. Each chiral ring with nitrogen. 1 nitrogen, second nitrogen, third nitrogen, and fourth nitrogen. The center atom is magnesium. And the phytal tail also present. This is the phytal tail. This is called as phytal tail. So this is the chloroplast structure. Phytal tail also present. Right. Pigment absorption this way. Let's see light absorption spectrum. So actually in atmosphere, the different type of lights are there, but in photosynthetically utilized light, VIBGR. What is VIBGR? VIBGR, here we can say this is this pattern, VIBGR, this wavelength pattern called as VIBGR. This is violet color, blue color, green color, yellow color, orange color, and red color. But it's not clear. Actually, this it should be clear red. right? So this is the wavelength and visible light, and this is called visible light. Apart from that, the another UV light, X-rays, and IR. So different type of lights are there in atmosphere, but they are not utilized for photosynthesis. But in photosynthesis, in photosynthetically, only the leaf can absorb visible light. Leaf can absorb the visible light. The visible light wavelengths also different. The first, for example, the violet color. The violet wavelength 380 to 450 nanometer. And next blue light. This blue light wavelength 450 to 495 nanometer. And next green light. Here it is. Green light. The green light wavelength 495 to 470 nanometer. And yellow light. This yellow light wavelength 570 to 590 nanometer. And orange 590 to 620 nanometer. And finally red 620 nanometer to 750 nanometer. So this is the visible light. In visible light, especially the photosynthetically active light, that is called PAR. Photosynthetically active light, that is called PAR, blue light and red light. I will explain later why it is only the blue and red photosynthetically active. The photosynthetic pigments observe only visible light, already told you, in order to get the photosynthesis. Hence, visible light is called as photosynthetically active, PAR light. But among this, the particularly only the blue light and red light. So the particular nature of light is usually expressed in statement that the light comes in quanta or photons. Actually, this light before the visible light, actually this is formed in the form of photons. Right? This photon energy, from the photon energy, only the visible light absorbs the plants. The energy in each photon is inversely proportional to the wavelength. Energy in each photon is inversely proportional to the wavelength. Inversely proportional to wavelength. That means if the wavelength is decrease, energy increase. So the violet and blue wavelength have more energetic photons than the orange and red ones. Right? So let's see some graphical representation. We can see here a different type of lights. Mainly, we can focus on the already told you blue light and red light. First, chlorophyll A and chlorophyll B. For example, chlorophyll A, just one. Right, sorry for disturbance. Chlorophyll A. This is A. Chlorophyll B. This is actually blue light. Blue. This peak is red light. Skip this carotenoids. Right here, you can see chlorophyll A. This is chlorophyll B. Green color chlorophyll B. Right? First, let's see the blue light. Blue light. Maximum absorption of blue light in chlorophyll A. Chlorophyll A, especially chlorophyll A. Maximum absorption of blue light. This is 400 nanometer, almost 435 to 40. So this maximum absorption rate spectrum 435. 35 nanometer. This is the maximum. Almost 435 to 450. That means. So maximum absorption of red light. Same the chlorophyll A. Same chlorophyll A. Maximum absorption of red light. So this is blue light. The same. This is the $600 73. 673 nanometers. 673 nanometers. So here chlorophyll A, one pigment, chlorophyll A pigment. The chlorophyll A pigment, maximum absorption blue light. This region blue light. Maximum absorption of blue light 435 nanometer to 450. In the same way, this chlorophyll A, this is the red light range. This red light range and this is the blue light range. In red light range, 673 nanometer. That is the maximum. Absorption only the maximum light 673 almost to about 700 nanometer. So let's see the chlorophyll B absorption rate. So chlorophyll B green color. Chlorophyll B is the green color. Chlorophyll A is the light yellow color. Chlorophyll A. Chlorophyll B is the green color line. If you see the green color line, the green color line in blue light. Green color line, if blue light, this is blue light, this is blue region, green color line, the maximum absorption rate at this region at 453. 453. The same light, red light range, $642 maximum. That means this is the photosynthetically rich 642. Right now, let's see the carotenoids. Another, of course, it's not important, but anyway, the carotenoids. Let's see in the blue color and red color. The carotenoid maximum absorption 449 nanometer 478 nanometer in blue light, and no absorption in red light. This is important. Is there any peak in red light? This is the carotenoid. The orange line is carotenoid. The carotenoid range almost in blue color. Only the carotenoid can absorb in blue light. In chlorophyll B, sorry, in carotenoid maximum absorption 449 nanometer. Or 472 nanometers to 478 nanometers in blue light. Only blue light. This is the carotenoid. The carotenoid can absorb only in blue light. Right? What about red light? In red light, is there any peak? Is there any peak in red line? No. So this is the absorption spectrum of the chlorophyll pigments. Once again, if you see only blue light, this range, just wait. Blue light range, red light range, right. First of all, in blue light, once again, don't get confused. In blue light, the chlorophyll A, the light yellow color chlorophyll A, green color chlorophyll B, this orange color carotenoid, right? Different peaks. Let's see the pigments absorption. First, chlorophyll A pigment. Chlorophyll A pigment, the variation in blue light and red light. First, the maximum absorption blue light. Blue light, chlorophyll A, 435 nanometer, almost 435, 440, 450. Not exceed 450. But rich is maximum rich absorption 435 nanometer, right? The same way in red light, this is this pigment, chlorophyll A pigment. This is the blue light range, and the same in red light. This is the red light range, 673 nanometer or 700 nanometer. But rich is 673 nanometer. Next, chlorophyll B, green color. Chlorophyll B, blue color 453, red color 642. If see the carotenoids, only blue light, but not in red light. So what you observed in this, the chlorophyll B, chlorophyll A, and carotenoids. First, if is chlorophyll A, when compared with chlorophyll B, especially in blue light, less observed. In blue light, only chlorophyll B, the most absorption wavelength, 453. And here, the same chlorophyll B, 642 in red light. So this is the absorption spectrum. Next mechanism. Already told you very beginning, in photosynthesis, 2 stages. Synthesis, light reaction. Second one is dark reaction. What happened in light reaction? I already told you. Let's complete again. Light. So already given. Light reaction. The end product of light reaction, NADPH formation and ATP formation. Right? These NADPH and ATP, these both are utilized for formation of glucose. Formation of glucose by next reaction, that is the dark reaction, by dark reaction. This is carried out by carbon assimilation. C3 cycle, C4 cycle, I will explain. But remember, 2 stages. One is light reaction, Second one is dark reaction. What happened in light reaction? In light reaction, just energy formed. This energy formed in the form of NADPH and ATP. I will explain clearly how the NADPH formed and ATP formed, right? So this utilize this assimilatory power, and finally the glucose formed and another stage, that is the dark reaction stage, right? Let's discuss first one by one. Before that, you should aware about this thylakoid structure. This is the thylakoid actually. If you see already we discussed earlier, if you see the thylakoid structure, chloroplast structure, chloroplast double membrane chloroplast. Inside the chloroplast, thylakoid is there, right? Thylakoid is also present. If you take the one thylakoid, each and every thylakoid is made up of double layer. This is called thylakoid membrane. This is thylakoid membrane, inner and outer. The inside, this is called lumen, thylakoid lumen. So this inner place, this one, thylakoid lumen. This thylakoid lumen, right? This is the membrane. This place. Membrane. And outside, so this is thylakoid, and outside the thylakoid place called as stroma. This region called as stroma. Now the light reaction takes place in this membrane, right? So light reaction takes place in this membrane, right? So in light reaction, in thylakoid membrane, phospholipids chain formation, like phospholipids membrane formed. In this mainly two stages, two lights, PS2, photosynthesis 2, and photosynthesis 1. This is acyclic reaction. Actually, light reaction, two stages. One is acyclic reaction, another one is cyclic. Acyclic means if the electron released from the reaction center and does not received again by reaction center, and finally this electron received by ATP or and formation NADPH, that is acyclic reaction. Cyclic reaction means in cyclic reaction involved only PS1. In cyclic reaction, the electron released from the PS1 reaction center, from the reaction center to ferredoxin, from the ferredoxin to again cytochrome, this reaction. Cytochrome, B6F, plastocyanin, again PS1. That means actually electron released from PS1 and again received by PS1. That cycle called as cyclic reaction. So anyway, the first let me complete about acyclic reaction. In acyclic reaction, first sunlight. Already just before we discussed about the light absorption spectrum in light, maybe chlorophyll A or chlorophyll B, blue light and red light in different wavelength observed. When they observe the wavelength in the form of photons, the form of photons, this energy, form of photon energy, first received by reaction center, PS2, photosynthesis 2, reaction center, chlorophyll A. So when the one photon energy received by chlorophyll A, automatically the loss of electron. So now reaction center oxidized. So this loss of electron again received by pheophytin, actually not written here, but remember pheophytin. Pheophytin received by pheophytin. What happened with the loss of electron? Again, actually this reaction center always filled with actual magnesium 12, right? Magnesium atomic electron 12, that always filled with electrons. Even number, that's why they always filled with electron. But what happened if one electron lost from the last orbital? So one single electron formed. This single electron always try to pair with another electron. So in the, for example, pair of electron, pair of electron, pair of electron. One photon observed, automatically this one electron, the one electron excited into outer orbital. Excited into outer orbital. From the outer orbital, it is received by pheophytin. The meantime, the water molecule again received photon light energy. As a result, split into two electron plus two protons of oxygen. Now this oxygen released into the atmosphere of oxygen released into atmosphere. Now this releasing electrons one by one, releasing electron one by one sent into the reaction center chlorophyll A. Right? This is the continuation process. First, actually, from the light photon energy, one electron lost. And again, this electron fulfilled by water electron. Right? Again, from the first, where the first electron pheophytin, with pheophytin. Now the pheophytin oxidized after loss of electron. From the pheophytin, again electron lost. Now this electron sent into the quinol cycle. I will explain clearly. Quinol cycle, sent into the quinol cycle. Right? Now the pheophytin again reduced with another electron. This another electron sent from the chlorophyll A, reaction center chlorophyll A. Now this chlorophyll A electron again filled with another electron. This is the cyclic process. Lost electron, gain electron, oxidoreductase process. Oxidoreductase, or this process called as redox process. In redox process, lost electron, gain electron. Lost electron, gain electron. Don't get confused. First, from the PS2 reaction center, chlorophyll A, lost electron. The lost electron and it is oxidized. And again, pheophytin receive the electron and reduced. Reduction. Gain of electron reduction, loss of electron oxidation, remember. So, to fulfill the orbital, the water molecule split into two electrons, two protons of O2. From this electron, one by one sent into the reaction center chlorophyll A. Hello? 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 Am I audible?
Yes, sir.
Right. Where we are? So always remember the first the filled electron, 12 electrons. So outer orbital, no lone electron. As it center one magnesium, as it electron efficiency 12. So always filled with two pair of electron. But when we received the photon energy, the loss of one electron, and this electron fulfilled by water electron. And it would be continued until formation of ATP, NADPH, assimilatory energy, right? Now pheophytin to electron sent into the quinol cycle. Here remember one interesting thing, for one electron, if one electron received by pheophytin, two protons received from stroma region. As a result, formed into phosphoquinone. This is phosphoquinone developed into phosphoquinone. From the phosphoquinone, again, the one electron sent into the cytochrome B6F complex. Cytochrome B6F complex. What about the protons? The protons sent into the lumen. This is for one electron. And the same way, if sent the another electron, the same goes to pheophytin. Pheophytin is a quinol cycle. If one electron received by quinol cycle, two protons received from the stroma region. As a result, quinol cycle, phosphoquinol formed. Now the another electron sent into the B6F complex, and this proton sent into the lumen, right? So from the B6F, these are sent into the plastocyanin. Before that, just let me explain brief about the quinol cycle, simple. Quinol cycle, plastoquinone, always with with one electron, already existed one electron, plus received one electron, right? In the same way, received how many protons? Two protons. And formed into phenol. PQH2, actually not here. PQH2, right? Plastoquinol formed. Just remember here this region. Just focus on this region. From plastoquinone in reverse. One electron loss, two proton loss. After loss, one electron, two proton, again converted into plastoquinone, right? Hello? Hello? Hello?
Yes, sir.
Right. So first, don't get confused. First, plastoquinone, one electron received. At the same time, two proton received from the stroma region, and plastoquinol formed PQH2. This PQH2, this PQH2 again convert into PQ. When it is converted into PQ, after loss of one electron and loss of two protons, right? This lossing electron received by this one electron loss. This lossing electron received by cytochrome B6F complex. Hello? What about two protons? What about these protons? Where they are? Hello? Hello? What about protons? I am asking about protons. This proton sent into the lumen. This is the lumen region. I already told you. This is the lumen region. The proton sent into the lumen region and again formed into PQ. And this is the cyclic process. Again PQ, the receive electron, receive protons, electron again the same way as usual sent into the B6F complex. The proton sent into the lumen. For one pair of electron, how many protons are sending into the lumen of thylakoid? For one pair of electron, one electron, two protons, pair of electron, four protons sent into the lumen, right? And here, how many protons are present inside the lumen? Through quinol cycle for one electron, two protons, and two protons, another electron, two protons. So total 4 protons for quinol cycle. What about water split? Through water split, 2 electron and 2 proton released. Through water split, 2 protons released. As a result, total number of protons, pair of electron, 6 protons. Total 6. This is for quinol cycle, this is for water split. So anyway, from the electron, from the cytochrome B6F, this electron transferred towards the plastocyanin. The plastocyanin existed outside a membrane towards the thylakoid lumen. This is loosely attached to the membrane. So this is the mobile carrier. Mobile carrier means it can move from the cytochrome B6F toward the reaction center 1. Toward the reaction center, PS photosynthesis 1. Now from the plastocyanin, electron sent into the photosynthesis 1. Now here the nanometer range 700 nanometer. Same here also light, red light at 700 nanometer. But here the red light 600 nanometer. PS2 600 nanometer. PS1 700 nanometer. Light received. When the light received from the PS1 reaction center, 1 electron loss. And this lasting electron fulfilled by plastocyanin. The plastocyanin already carry the electron very sooner if the loss of electron from the reaction center again it sent to the reaction center. The plastocyanin gives to the PS1 photosynthesis 1 reaction center. Now this releasing electron received by ferredoxin, from ferredoxin to this electron received by NADP. NADP received by NADP. This NADP along with electron and received some protons also from the stroma. As a result, developed into NADPH2. Developed into NADPH2 assimilatory power. Already told you very beginning, through light reaction, assimilatory power formed NADPH2 and ATP. Let's see now how ATP formed. Now ATP formation called as phosphorylation. So this process called as photophosphorylation. Photophosphorylation. What is photo? Photo means light. Phosphorylation means phosphate formation. Let's see the photophosphorylation. In photophosphorylation, actually in where is cursor? Just wait. Right. In lumen, total how many protons are formed? 6 protons formed. 2 from water, 4 from quinol cycle. Actually, these protons always try to move into the stroma because these protons already already existed inside the stroma region. That's why they wants to move into the stroma region. But this membrane, this thylakoid membrane, impermeable to move towards stroma region. not allow this membrane, not allow into the stroma region. That's why it takes another way. That's why this choose the coupling factor. F0, F1 coupling factor, F0, F1 coupling factor. This is head region, this is head region, and this is tail, right? So these photons can move through this coupling factor. For a year, through this coupling factor, F0, F1 coupling factor, for three protons, three protons move from the straight. For three protons travel through the coupling factor, these protons one by one, not enter through these photon one by one, travel through the coupling factor. When one proton travel through this head region, automatically this head region rotate. That finally, if three protons are travel through this coupling factor, head region factor, one ATP formed, one ATP generated. Like this, how many protons are formed in this lumen? Total 6. For 3 protons, 1 ATP formed. Then how many ATP formed for 6 proton? For 6 proton, if travel through this coupling factor, 2 ATP formed. So ATP also ready, ATP formation also clear. NADPH formation also clear. So NADPH formed and ATP formed. And this is called assimilatory power. Already told you very beginning, light reaction means utilize light and formation assimilatory power. This why it is importance formation of assimilatory power, where it is required. This power where it is required. This formed assimilatory power NADPH, NADPH2 or NAATP utilized for formation of glucose. This reaction called as dark reaction, right? So this is the acyclic right reaction and acyclic phosphorylation. Let's see the cyclic reaction. The simple with the cyclic reaction, if you see the cyclic reaction, first only reaction center, this only involved reaction, acyclic photophosphorylation only involved reaction center 1, not reaction center 2, only involved reaction center, right? From the reaction center, after received photon energy, electron released. This electron received by pyridoxine. Actually, from the pyridoxine, it to be sent to NADPH, but instead of that, this electron in cyclic, I am talking about cyclic photophosphorylation. Instead of that, this electron received by cytochrome B6F. Cytochrome B6F. Sorry, not cytochrome B6F. Electron received by phenol cycle. From phenol cycle to same B6F. B6F to plastocyanin. Plastocyanin to again reaction center. That's why it is called as a cyclic, because now the electron released from the reaction center and received by the same reaction center. That's why it is called as a cyclic reaction. What happened through the cyclic reaction? In the same way, when the electron received by phenol cycle, 2 protons received. For another electron, another 2 protons received. That's why through this phenol cycle, 4 protons are formed. 4 protons are formed. These 4 protons, if travel through the F0Fn complex, 1 ATP formed, extra 1 ATP formed. So this is called cyclic phosphorylation. This is called cyclic phosphorylation. So this is only blue lines. Remember this blue lines. PS1, pyridoxine to phenol cycle to cytochrome B6F to plastocyanin. Right. This is cyclic reaction. Right. Any doubt in this? Hello? Clear? Light reaction clear? Simple and light reaction. You must have confused. Just wait. Thylakoid membrane. PS2, photosynthesis 2, photon energy, utilize photon energy, lost 1 electron. This electron received by pheophytin from pheophytin to phenol cycle, plastocyanine. If plastocyanine received 1 electron, this is stroma region. Sorry, forgot stroma. Thylakoid lumen. From stroma region, 2 protons received. 1 electron, 2 protons. Convert into PuQH2. Again, this plastophenol again convert into plastoquinone after loss of electron and loss of protons. Simple. 2 protons. No, actually after loss of electron from the reaction center to lone electron formed. This lone electron again formed as a pair electron by water split. Say water molecule. Two electron plus 2 protons of O2. Now this electron sent to the reaction center. Continuation. This is a cycle, like a cycle. From reaction center pheophytin to plastoquinone. While the first received by plastoquinone, same 2 protons received by plastoquinone, and again formed as the plastoquinone. The same way, again it is converted to plastoquinone after send the electron, send the protons to the lumen. Here these electrons sent to the 1 by 1 cytochrome B6F. From this electron sent to the plastocyanin. This is the mobile carrier. Plastocyanin. Loosely attached to the membrane. Now this electron from the plastocyanin towards the reaction center 1. Before that, actually from the reaction center at the 700 nanometer wavelength, photon energy received, as a result loss of electron. Now this electron fulfilled by plastocyanin carrying electron. From this electron sent to pyridoxine to Na Tp plus. This Na electron 1 by 1 received 2 electron plus 2 protons already present in the stroma region. As a result, formed into Na DPH2. Right? So one assimilatory power formed, Na DPH2 clear. Formation Na DPH clear. And another assimilatory from ATP formed. Now this actually, these protons after split 6 protons. This is from water molecule. This 4 from the phenol cycle. From the phenol cycle 4. These always try to move into the stroma because they are coming from stroma. That's why they always tries to move into the stroma region. But it is act as this membrane act as impermeable membrane. That's why this is take another way, that is the coupling factor. F0, F1, add region, tail region. So now these protons travel through this. For 3 protons, ADP utilized phosphate convert into ATP. For 3 protons, 1 ATP formed. Like that, here formed 6 proton. So if the 6 proton travel through the factors, 2 ATP formed. Like this assimilatory power formed. Now this assimilatory power NADPH to NADPH2 plus 2 ATP utilized for formation of carbohydrate in the for presence of carbon dioxide, water molecule. With help of this energy reduced as a C6H12O6. C6H12O6 and also release oxygen. During photosynthesis, release oxygen, already told you. So oxygen released in light reaction here. Right? Tomorrow we will discuss about the light reaction. Light reaction, carbon assimilation, C3 cycle, C4 cycle. Right? Already asked you about the group. Are you in group or not? Are you maintain a group? Hello?
Yes, sir. Yes, sir.
Please add my number in our group.
Okay, sir.
Already told you in my first class. So that I sent some videos. Okay?
Yes, sir. Okay, sir. Okay, sir.
Correct.
Today definitely I'll add it.
Okay, okay. Thank you. Thank you. And we will discuss tomorrow about carbon assimilation, dark reaction. Okay? Today class clear? Any doubt in this?
No, sir. Yes.
Right.